Do you or a family member suffer from Huntington's disease? AMT-130 is the first one-time gene therapy under current clinical trials for the treatment of HD. It is targeted toward the caudate nucleus and the podomy deep brain structures that are responsible for processing information and are affected first by HD. AMT-130 is administered through a small catheter that is inserted into your brain. 3 infusions on each side of the brain are performed over an 8 to 10 hour period using a magnetic resonance imaging or MRI scan to monitor the delivery of AMT-130 to the deep brain structures. AMT-130 diffuses throughout the caudate nucleus and the podyne and delivers the therapy that lowers the production of Huntington proteins. Research shows that AMT-130 spreads throughout the rest of the brain, including to the cortex and other regions affected by HD. How does AMT-130 work? AMT-130 is delivered into brain cells using tiny carriers called AAV vectors. The AAV vectors move into the cell's nucleus, which is like its control center. Inside the nucleus, AMT-130 doesn't change the cell's DNA but sends instructions to target and reduce the production of harmful proteins related to Huntington's disease. By reducing these harmful proteins, AMT-130 aims to slow down or stop the progression of Huntington's disease. New treatments have to go through several phases of clinical testing before they are made available to patients. The first phase is the preclinical phase. Here, researchers perform laboratory and animal tests to assess the safety and effectiveness of a new treatment before it is tested on humans. Next, we move on to phase 1, where researchers test the treatment on a small group of people to assess its safety, determine safe dosage ranges, and identify side effects. With relative success and safety, the treatment moves on to phase 2, where researchers test the treatment on a larger group to further assess the safety for a specific condition. AMT-130 is currently undergoing phase 2 clinical trials on patients with Huntington's disease. To be eligible for the study, you must be patients with a definitive clinical diagnosis of early manifest HD, males and females, between the ages of 25 to 65 years old, and have a CAG repeat expression equal to or greater than 40. This is the initial human study of AMT-130 gene therapy, which has shown general safety in animal tests and is based on animal dosing. While some AAV vectors like those used in AMT-130 are approved and considered safe, the risks include the permanent nature of gene therapy, unknown effects of altering Huntington protein levels, potential ineligibility for future gene therapies, and unforeseen side effects, including serious reactions. Delivery to the brain introduces additional risks related to general anesthesia and microcatheter use. To join phase 2 of our clinical trial for AMT-130 gene therapy for Huntington's disease, please visit www.clinicaltrials.gov.